In this video, we're going to have a look at gene mutations and how they can result in cancer. So before we start, we'll do a bit of a review, a bit of a revision on your current knowledge and understanding of mutations. Adding to that, so in your current understanding, you should know about substitution, addition, and deletion types of mutation, and we're going to add to that duplication, inversion, and translocation mutations. Then lastly, we're going to look at how mutations to certain genes can lead to cancer. So, a mutation can be defined as a change in the amount or the structure of DNA. A gene mutation is a change to the nucleotide base sequence, and this is caused by errors in the replication of DNA. Chromosome mutations are a change in the number or the structure of chromosomes, and this is caused by errors in cell division. So, for example, non-disjunction during meiosis, and specifically the non-disjunction of chromosome number 21 could lead to Down syndrome. That means having 3 copies of chromosome number 21, sometimes referred to as trisomy 21, leading to Down syndrome. But in this lesson, we're going to concentrate on gene mutations, so those changes to the nucleotide base sequence. So, what causes mutations and what are their consequences? Firstly, it's really important to remember that mutations are spontaneous. They're happening all of the time, and there is nothing specifically which causes them to happen. However, there are chemicals or agents or factors which will increase the rate at which those spontaneous mutations take place, and we call these mutagenic agents. So, for example, X-rays and other ionizing radiation, such as UV light, and some chemicals such as benzene are all examples of mutagenic agents which increase the rate at which mutations take place. Some mutagenic agents can be further classified as carcinogens. So these disrupt the genes for proteins involved in the cell cycle regulation or involved in mitosis. And the disruption of those genes for those proteins involved in cell cycle regulation can lead to the development of tumours. So carcinogens are a sort of subset of mutagenic agents which can lead to the development of tumours. Mutations are not always bad. There's a little bit that's been cut off the bottom there. And in fact, they are important for natural selection, for the production of new alleles, which may have a beneficial effect, may increase the reproductive success. So here we can see a mutation which creates variation. So obviously we know that natural selection depends on there being mutation present in a population. Unfavourable mutations tend to be selected against, okay, because they will lower the survival chances and the reproductive rates of those individuals in the population with the unfavourable mutation. So they are selected against. The favourable mutations, those ones that give the individuals possessing those alleles an increased chance of survival, mean that those individuals with the favourable alleles are able to reproduce more often and for longer, and therefore those favourable alleles will increase in frequency, and that's demonstrated in this diagram in the top left. So, errors in replication. Let's just recap on 3 that we looked at in booklet 7 at the end of the first year. Each of these, substitution is referred to here as an insertion, but our specification means an addition or a deletion. Each of these is what's called a point mutation, so a change in the nucleotide sequence of a gene, and it occurs during DNA replication. So, in a substitution, as DNA is replicated, one nucleotide may be exchanged for another. This can happen if the base is paired wrongly due to one of those mutagenic agents, such as ionizing radiation or chemical damage. Okay. Here we can see the incorrect base has been substituted. Okay. When the gene containing the error is replicated at the next in the next cell cycle, at least one copy will retain that error, and the incorrect base will be paired with its complementary nucleotide, and this is what will complete the mutation. So substitution mutations tend to only cause minor problems. Here we can see that this triplet code eventually coded for the wrong or a different codon on the mRNA, which would be complementary to a different anticodon on the tRNA, and cause the change in the amino acid sequence, i.e. a change to the primary structure. And we'll have a look at what effect that change to the amino acid sequence in the primary structure has later. Okay. In addition mutations, referred to here as an insertion, as the DNA is replicated, the DNA polymerase may slip and repeat a base or sequence of bases, and this causes the addition of one or more extra nucleotides to the new strand. You can see here DNA polymerase slipping and adding an additional nucleotide to the new strand. In the next stage of replication, then, the extra base is matched with its complementary base pair, forming a new base pair and changing the nucleotide base sequence. The significance of this is that all of the base sequences subsequent to the mutation, so after that mutation, will now change. We call this a frame shift, where all of the triplet codes after that point of mutation will change, and this will change the or lead to a considerable change in the amino acid sequence of the protein. So a major change to the primary structure of the protein. Lastly, a deletion. So as the DNA is replicated, the DNA polymerase may slip again, overlooking a base or a sequence of bases, causing one or more nucleotides to be missed from the new strand. I can see that there. Okay, so here is the new strand being produced on the right there. We can see one of the bases is missing. Okay, so in the next replication then, one of the daughter strands will be missing one or more base pairs, and again this results in a frame shift. So again, all of the triplet codes after that point of deletion, after the mutation, will change, and subsequently or consequently all of the amino acids after the point of the mutation will change. So again, considerably changing the primary structure of the protein. The consequences of a mutation can be threefold. There may be no change to the amino acid sequence. So, due to the degenerate nature of the triplet code, where more than one triplet may code for the same amino acid, a change to the triplet code, a change to the nucleotide base sequence, may result in one of those other triplet codes coding for that same amino acid, and therefore there will be no change to the amino acid sequence, no change to the primary structure, and no change eventually to the tertiary structure. Alternatively, the change to the nucleotide base sequence could result in the introduction of a stop codon, and this would mean that protein synthesis was prematurely terminated. The polypeptide is shorter than had been intended, and obviously that would result in a non-functioning protein. Or a different amino acid could be added to the polypeptide. This would change the primary structure of the polypeptide, and a change to the primary structure may result in the formation of bonds, so those hydrogen or ionic or disulfide. I should say disulfide in different positions. If those bonds form in different positions, that causes a change to the tertiary structure. And a change to the tertiary structure may result in a protein that functions differently, or a protein that doesn't function at all. So, this change to the change to the nucleotide base sequence that results in a different amino acid being added to the polypeptide equates to a new allele, which increases the genetic variation. In some cases, that might be an advantageous change. Okay, so the new allele may provide the individual with a greater survival chance, increase the reproduction of that individual, meaning that eventually the allele frequency increases. Okay, that's the concept behind natural selection, which could eventually lead to evolution. Alternatively, that new allele could be detrimental and disease-causing, which would perhaps lower the survival chances of the recipients of that new allele. Okay, here we can see a substitution where adenine in the DNA strand has been substituted for guanine. Now, just a reminder, but due to that degenerate nature of the triplet code, where just remind ourselves that more than one triplet can code for the same amino acid. Here we see this triplet, GAA, guanine, adenine, anidine, coding for glutamic acid. A substitution of adenine for guanine, due to that degenerate code, this also codes for glutamic acid, and therefore the primary structure, the sequence of amino acids in the polypeptide, remains the same. It would fold into its secondary structure and its tertiary structure in the same way, and those hydrogen ionic and disulfide bonds may form in the same place. So there will be no change to the protein produced. Here we can see that adenine again is substituted, but this time for a cytosine. Now, glutamic acid is replaced by aspartic acid. So here we have a change to the amino acid sequence. I'm sorry, I don't know why it's doing these funny lines. If there's a change to the amino acid sequence, those hydrogen ionic disulfide bonds may form in different positions, which would change the tertiary structure, which would result in a protein that functions differently or perhaps doesn't function at all. So a change to the amino acid sequence in the primary structure may cause hydrogen bonds in the secondary structure to change position. Okay, this may change the secondary structure, perhaps those alpha helix or the beta pleated sheets may form in different positions, which would affect the shape of the protein. In the tertiary structure, again, hydrogen bonds and ionic bonds and disulfide bonds may form in different positions, which would change the three-dimensional tertiary structure of the protein and may perhaps cause a change to the function of the final protein. An example of a mutation which leads to the change in the primary structure and the subsequent change to the secondary and tertiary structure is sickle cell disease or sickle cell anemia. In this disease, the normal adult hemoglobin, HB, hemoglobin A, contains a glutamic acid at the 6th amino acid. In the mutated hemoglobin S, that glutamic acid, coded for by guanine, adenine, guanine on the DNA strand, the nucleotide adenine is replaced by a thymine, This GTG codes for valine rather than glutamic acid. This produces a mutated form of the beta globin molecule. So each hemoglobin molecule consists of 2 alpha and 2 beta globins. The beta globins in individuals with hemoglobin S is different. It has a valine rather than the glutamic acid. At low partial pressures, that the mutated beta globin causes the red blood cells to collapse into this sickle shape. They are less elastic, they're more likely to stick together and cause blood clots. Also, to remove those sickle cells, these red blood cells undergo hemolysis. So they break up and are removed. And although the bone marrow tries to compensate for that by producing more red blood cells, they're not replaced at the same rate that they're lost. Hence we get this anemic condition where the blood is able to carry less oxygen. So the other types of gene mutation are a duplication. This is where one or more bases is repeated. You can see here that these two, the cytosine and guanine, here are repeated again. This one again causes a frame shift. Okay, so after the point of mutation, all of those amino acids in the sequence will be changed, which could be fatal, or at least cause the production of a non-functioning polypeptide and protein. The next is inversion, where a sequence of bases is reversed. Here we can see the TAG, CAG, C. That has been reversed, it's been switched over. TCGA, CGAT. Okay, it is the reverse of this first sequence. Okay, and that would change the order of the amino acids. Depending on the length of the DNA which is reversed, that will decide the amount of change to the amino acid sequence, the primary structure of the protein produced. And lastly is translocation. This is where a sequence of bases is moved from one part of the genome to another, i.e. from one chromosome to a different chromosome altogether. Okay, so mutations then may be related to cancer. Let's have a look at how that might happen. In the control of cell division, there are genes involved. So, during each cell cycle, cells will divide to replace dead cells or damaged cells, and the rate of that cell division is controlled by two groups of genes, or two genes which can be classified as either being proto-oncogenes. Now these proto-oncogenes will stimulate cell division, or tumour suppressor genes, and this group of genes will slow or stop cell division. Mutations to proto-oncogenes and tumour suppressor genes have been found to be the cause of many types of cancer. A mutation to the proto-oncogene changes that proto-oncogene into a gene we call an oncogene. That oncogene leads to excessive, uncontrolled and chaotic cell division. The cells that divide may stick together to form a tumour, and then the tumour may break apart or metastasize and move to other parts of the body to infect or to invade different tissues and different organs around the body. Here we can see a scanning electron micrograph of lung cancer during cell division. A mutation to a tumour suppressor gene leads to that tumour suppressor gene becoming inactivated. So usually a tumour suppressor gene would slow down the rate of cell division as a way of regulating that cell division. If the mutation to the tumour suppressor gene results in that tumour suppressor gene becoming inactivated, again, cell division would continue, but in an unregulated way. It may increase in rate. That cell division may be chaotic again. It's essentially uncontrolled, and that leads to the development of tumours. Just to repeat, those tumours then may break apart or metastasize. They may move and invade other tissues close to the original site of the tumour or elsewhere in the body as they're carried by the lymphatic system or the blood to other regions of the body.